Good morning, everyone. So we had started studying genetics. Let's start with this one UPSC previous year question. RNA interference or RNAi technology has gained popularity in the last few years. Why? So UPSC asked this question and they've given you 4 options here. Okay. The answer I'm getting online is also A. First one is it is used to develop gene silencing therapies. So this is definitely correct. We had studied about gene silencing technologies, and in that we had learned RNA interference as the post-transcriptional gene silencing technology. It can be used in developing therapies for treatment of cancer. So yes, if there is a cancer which is related to a gene defect, we can use this kind of a technology to develop something to suppress that gene which is responsible for causing cancer. So that kind of technology could be developed, and with that we get our answer, which, like you've said, is going to be A. We are going to go through the other 2 options also. It can be used to develop hormone replacement therapies. Not really. If you have to replace a hormone or change a hormone or add an hormone, you will need to add something, okay, to give that hormone. Suppression will not really work for that. And the last option, it can be used to produce crop plants that are resistant to virtual pathogens. So we can definitely, whenever there is resistance, or if you want to create that special property that a crop should be able to survive a pathogen, we can silence the part which is being attacked by the pathogens, and through that kind of a recombination this can happen. But this is in any case automatically confirmed from your option A. So A would be the right answer here. Now we had studied about how chromosomes are there, how they are working, how genes are working, how inheritance is happening in one generation to the next. We are going to start talking about genetic defects now, or genetic abnormalities. The system of inheritance cannot always be perfect. There is a possibility that there can be some variation, some alteration in that. And when that happens, the person will have some problem, the person will have some disease. There are many ways in which genetic abnormalities can come up. The first case which we are going to consider is when you have an abnormal number of chromosomes. In all species, we have studied that the number of chromosomes has to be maintained. You have the same number of chromosomes in every person of a particular species. What is the number of chromosomes in human beings? 46, right? If there is a situation जहाँ 46 के अलावा कोई भी और number हो, that is going to result in the form of a disease. So let's take an example. These are the chromosomes of a person. We saw a diagram like this last time also, okay, when we studied homologous chromosomes. Now, in this, we had studied that we have 23 pairs of chromosomes. In every pair contains 2 chromosomes. Now, if in any of these pairs there is an extra chromosome or there is a chromosome less, both options are possible. In that case, we will have some disease. And the disease will depend on which chromosome is extra or deficient. This is the most common example of such a chromosomal abnormality. It is called Down syndrome. You might have heard about it. Down syndrome is because of presence of an extra chromosome number 21. So like all these pairs, chromosome number 21 was also supposed to be having just 2, but this now has 3 chromosome number 21s. So because of one extra chromosome number 21, the person has Down syndrome. बाकी भी कोई भी क्रोमोसोम एक्स्ट्रा हो सकता है, but that will be a different disease. Very sorry. This Down syndrome is specific to chromosome number 21. For any other chromosome, there will be some different disease. Okay. People with Down syndrome or children who are born with Down syndrome, you can see them and identify them if you've like seen their pictures. They have a very unique characteristic appearance. They have a small rounded face, the mouth is open. Okay, that kind of an appearance is there. So you can just write this example, Down syndrome. Down syndrome, and it is also called trisomy of 21. Down syndrome, trisomy 21, that is presence of one extra chromosome number 21. One extra chromosome number 21. The child has a small rounded head, partially open mouth, and mental retardation. The next example which we are going to see is going to be on the sex chromosomes, and this is called Klinefelter's syndrome. Klinefelter's syndrome. In this, you have a male who is normally supposed to have X and Y chromosome, but in addition, he can have one extra chromosome, one extra X chromosome. And because of this man will have development of feminine characteristics. Sparse body hair, enlarged breasts, high-pitched voice, all the features which are the sexual characters of female, this man is going to have because of the abnormality in the number of chromosomes. So with Klinefelter syndrome, please write XXY, and then you can write this is a male This is a male with development of, this is a male with development of feminine characteristics. A male with development of feminine characteristics. Next is Turner syndrome. In this, you have a female who has one less chromosome. So the female, if you look at her sex chromosome, she was supposed to have two X's, but she has only one. The other place is vacant. It's usually written as 0 or O, XO we call it. So there is one chromosome less here. This female will be sterile. She will not be able to reproduce. So for Turner syndrome, please write XO. Turner syndrome, XO, and sterile female. This is a sterile female. Now, if I ask you what is the total number of chromosomes in Turner syndrome, what will that be? Hmm? 45. Everything else is fine, just one X is less. Okay, so it will be 45. Klinefelter's 47. Okay, everything else is the same, everything else is fine, just one extra X chromosomes. What about Down syndrome? 47 again. Okay, everything else is fine, just one extra chromosome number 21. So 47. Okay. Now, as of today, the situation is that for such abnormalities in the number of chromosomes, we really don't have any solution. Okay, there is no way of correcting it, there is no treatment, nothing like that. The person has to adapt and live with the abnormality which he is born. The only thing which can be done is that such a condition can be identified very, very early. Very early at the stages of the embryo, before the fetus is fully developed. At that stage, you can identify any such chromosomal abnormality, and then there would be a choice if this embryo should be allowed to grow or not, if this pregnancy should be continued or not. That option is available. But other than that, if this child is born, then there is nothing else which can be done. Okay. Okay. Now there can be another kind of abnormality. This was the abnormality where the whole chromosome is involved, but we can have defects in individual genes also. So one particular gene which is responsible for one function, that can be defective. Okay. When we look at the types of chromosomes, just like in the number, we had an abnormality in sex chromosomes also, and we could have an abnormality in the number of autosomes also. Same logic goes for gene defects also. So the genes present on any of these chromosomes could be defective. Sex chromosome का gene भी defective हो सकता है, autosomes का gene भी defective हो सकता है. Okay, so depending on what gene is going to be defective, we are going to have different types of diseases. First, we are going to talk about autosomal diseases. And the most characteristic example of this is sickle cell anemia. One of the most common gene defects. What is anemia? Deficiency of iron could be one thing. Deficiency of hemoglobin. Yes. So it's basically the hemoglobin which is less, and that hemoglobin could be less because of any reason. One of those reasons could be iron is less. That is going to be the reason for less hemoglobin. Now this is the situation of the cells of sickle cell anemia. Okay. If you look at the cells, these disc-shaped cells, these are the normal red blood cells, disc-like, or we call them biconcave in shape. ये normal appearance है red blood cells की. But because of a defect in the gene responsible for this shape, their shape becomes like that of a sickle. Sickle is a farm instrument. It's shaped like this. Okay, somewhat like this. And it is because of this shape that the name sickle cell anemia is there. Now in this shape, the problem is that it is very difficult to accommodate hemoglobin inside the red blood cells. Hemoglobin is present in the red blood cells. लेकिन ये shape ही ऐसी है कि इसमें अच्छे से hemoglobin is not able to be present. Because of this, there is an inherent genetic cause of having less hemoglobin just because of this shape. And whenever we have less hemoglobin, we say that condition is anemia. This anemia, however, is genetically determined. It is because of the defective genes which are present. Okay. There is another interesting thing associated with sickle cell anemia. If you remember, we had studied about malaria, and the organism causing malaria, Plasmodium, it reproduced in the red blood cells. Okay. Now, the sickle cell anemia shape, or the sickle cell shape, like it doesn't allow hemoglobin, it does not allow Plasmodium also to go inside. So people who have sickle cell anemia, they are automatically protected against malaria. So in any region where there is a higher incidence of malaria and people die because of malaria, we usually find an incidence of sickle cell anemia also. Okay, like countries in Africa where malaria is very prevalent, and even in India, tropical, subtropical countries, they suffer from the vector-borne diseases. So in India also we find that there are regions where sickle cell anemia is prevalent, and at the same time malaria is also prevalent. Now let's see how the inheritance or the gene pattern works for sickle cell anemia. Suppose HB is my normal gene. I'm just trying HB to connect it to hemoglobin, right? This is just for representation. Just like you had G for green color of P, A for A blood group, HB I have written for the normal gene responsible for this. HBS is now the defective gene. Okay. Now for inheritance, we have to see combinations. Like it was GG and GY and YY. So here also we will try and create all the combinations and see what's happening. So if a person has both his genes normal, he's obviously going to be normal. If the person has both defective genes, he's going to have the disease, he'll have sickle cell anemia. Okay. But there is a possibility of a heterozygous form also. HB, HBS. right? This combination, like we had GY. So ये heterozygous combination भी बन सकता है. And here, how do we decide that if this is going to be normal or diseased? Dominant, yes, very good. So when we had GY, we had to see which is dominant. अगर green dominant है, plant green होगा. ठीक है? In this case, when it comes to genetic defects, what we observe is that your genetic diseases, they are mostly recessive in nature. When I say recessive, that means the normal gene that is dominant and the defect or the defective gene that is recessive. There is a possibility that defective gene dominant bhi ho jaye, but that is very, very rare. For most of the diseases which we know, the defective gene is going to be recessive. So as a rule, that's how we consider it, because the common examples have the defect being recessive. Now, if this is the case, HB, HBS, will it be normal or diseased? Normal. So this person is going to be normal, because he has a normal gene which is going to dominate over the defective gene. Just like GY was green. GY was not a mix of feature or was not yellow, it was green. So यहाँ पे भी HB, HBS is going to be normal. However, there is a possibility that this normal person can transfer the disease to the next generation. If you have two such people who are carrying defective genes, they're both normal, they don't have sickle cell anemia, but when they have children, there is a high possibility that their children, they can give their defective genes, both of them can give it to the child, and the child can have sickle cell anemia. Now, because of this, even when they are normal, they can transfer the disease. We use a word to describe this, which is a carrier. When we studied different diseases, there we said that there is a vector which acts as a carrier of a disease. Do you remember that? जैसे मलेरिया की बात की है. So there is Anopheles mosquito which is carrying plasmodium from one person to the other, and that's how it's causing malaria. But Anopheles mosquito in itself does not get malaria, even when plasmodium is in its body. Same thing is happening here. These people who are going to be carriers, they don't have the disease in themselves, but they can transfer the disease to the next generation. So we use the word carrier for them. जिसको disease है उसने तो transfer करना ही करना, ठीक है? But these are people who don't have the disease, but they can still transfer the disease. This makes them carriers of the disease. And this is the reason, or this is the logic behind genetic defects in places where there is a lot of endogamy. That means marrying within close groups, marrying within families. Right? You have, the European royal families. So they have this idea of purity of line and all of that. So they marry within linked groups. And in that, there is a possibility that two of these defective genes could come together and genetic defects might show up. जैसे आप सोचो कि somebody has sickle cell anemia, so there is a possibility that he will pass that defective gene to most of his children. On subsequent generations, that gene will continue to be inherited, even if it doesn't lead to more diseased people, but genes to transfer hongi. So there can be a lot of people in that family who will have this gene combination, the heterozygous form. Now, if two such people get married and choose to have a child, there is a possibility that there is a higher possibility that these two defective genes can come together, as opposed to if this person would have married somebody else from another family जिस family में sickle cell anemia का defect नहीं था. So उसमें chances of a child having sickle cell anemia is reduced. Okay, this is one of the reasons for transmission in India. So sickle cell anemia has been in news. You'll get to read about it in current affairs, and I think June या July किसी month में है भी ये. So the government has started a program where they want to give counseling for this disease. They want to have earlier screenings before marriage and all of those things. The problem is that sickle cell anemia is very prevalent in tribal communities. Okay. Why in tribal communities again? You have to think of this, that they are smaller groups and marriages are within that groups. So continuity of gene defect will be there. The other thing is malaria also being responsible somehow for this. Because in these places, because of less access to awareness and medical services, prevalence of malaria is also high, and because of malaria's prevalence, that natural advantage makes sure that sickle cell anemia will continue. Think of this like suppose there is a population जहाँ पे बहुत ज़्यादा मलेरिया है, people are going to die because of malaria. And why sickle cell anemia also keeps continuing there is because the people who don't have sickle cell anemia, they get malaria and they die. So the only people left to reproduce and produce more children would be the one who have sickle cell anemia. ठीक है, तो इस तरह से वो कंटिन्यू होते रहते हैं, right? For such kind of diseases where just one gene is defective, we now have treatment options which we are exploring, where we'll have the possibility of changing that one gene. We'll talk about that technology also later today, right? So abhi wider scale पे use नहीं कर रहे हैं, but experimentally it has been given clearance that we can treat, we can remove this defective gene. ठीक है? Okay. Now this kind of a gene defect can also happen on a sex chromosome. The next thing we have to talk about is that. Before we go into that, we have to go back and look at the sex chromosomes first. Just look at this image where you have X and Y chromosome. Can you see there is a difference in the size of X and Y? Yes, Y is smaller than X. Okay. Now because Y is smaller than X, Y also has less number of genes. And if there are less number of genes, the chances of that being defective is also less. So when we talk about sex-linked gene defects, we are mostly talking about gene defects related to the X chromosome. Y पे भी हो सकते हैं, but कम होते हैं. कम genes हैं, तो chances of having a defect is also less common. So your sex-linked diseases, the most common examples are the X-linked diseases. The common examples here are hemophilia. So hemophilia is a disease of clotting of blood. People with hemophilia take a lot of time to form a clot. So even with the most minor injuries, they continue to bleed. So there's a lot of blood loss, even with the slightest of injuries. Queen Victoria had hemophilia, and this defective gene continues in her family. So this is also called royal hemophilia, okay, or royal disease. Another example is color blindness. Color blindness is when people have an inability to distinguish between primary colors. Now, X and Y chromosomes, they have different genes. And this defect, or the gene which is responsible for this, that is present only on the X chromosome. Okay. So let's see what's happening here. If we look at the normal people or normal genes, we are writing it as X. The normal people, their composition would be XX, XY. Okay, male and female. The defective gene which is present on X chromosome. So if I'm talking about hemophilia, I'm going to be writing it as XH. X chromosome carrying a defective gene for hemophilia. Now we are going to see the combinations. See, inheritance of these individual genes works the same way as Mendel's laws. So जो Mendel का inheritance pattern था, that is exactly going to be followed here. And we have many applied questions on such things. So for those questions, you'll have to work out that Mendel's probability and find out what is happening. Okay, so normal female, and let's write XH, XH, a female with 2 defective genes on X chromosome. This female is going to be diseased. Another situation where one X chromosome gene is normal, one is defective. So in this case, the female is going to be diseased female. This is going to be normal female, but she is also going to be the carrier. This disease is also recessive. Most of your gene defects, they are going to be recessive. So ये जो defective gene है, ये है recessive, और dominant है normal gene of the X chromosome. Now the case of the boy, of the male. This you have to understand very, very carefully, because what is happening is male के दोनों chromosomes अलग अलग है, दोनों पे अलग अलग genes है, ठीक है? So they really don't have a dominant recessive kind of interaction amongst themselves because they are different. जो gene ये disease cause कर रही है, that is present only on X chromosome, not on Y. उसका normal variant भी Y पे नहीं होता. So with just one X chromosome which is defective, the male in this combination will get the disease. So males, in case of sex-linked gene defects, cannot be carriers. They will either have the disease or they will be normal. For surviving, for being normal, you need one normal X chromosome. Okay, that is a condition. यहाँ पे मेल में एक ही X chromosome है, that also has a gene defect. So उसको counter करने के लिए, उसको dominate करने के लिए, you would need another X chromosome. Y will not work. Y as such is a smaller chromosome with very less number of genes. So in this combination, the male is going to be diseased. ठीक है? Okay. Now there are many questions on this in many exams, including UPSC. So we have this one question which you just try and solve. You'll also get a little clarity on how much you've understood. this will follow Mendel's inheritance, so you can make a cross and try it. D, okay. In the context of genetic disorders, consider the following. A woman suffers from color blindness while her husband does not suffer from it. Okay, अच्छा ये लिख लेते हैं पहले. There is a woman who suffers from color blindness. So if a woman is suffering from color blindness, her genotype will be carrying two defective genes for color blindness, XC, XC. That's how she has the disease. While her husband does not suffer from it. So husband is normal. Okay, let's make a cross and see what will happen. So first we'll make the gametes. And now we will check for all combinations. So X, XC, again X, XC, then XCY and XCY. So we have the composition of the girl child. This is what the girl child or XX is going to be like. One gene normal, one defective. Will the girl be normal or diseased? Normal. क्योंकि एक normal X chromosome है to dominate over the defective gene. And XCY, this is the composition of the boy. In both cases it's the same only. So this boy with XCY, one defective X chromosome and one Y chromosomes, will he be normal or diseased? Diseased. इसके पास एक ही X chromosome है, एक ही defective है, so he'll have the disease. So that means in all cases, the female or the girl is going to be normal and the boy is going to be diseased. So you have to find an option which satisfies this. The answer I'm getting is D. Both children suffer? No. Daughter suffers? No. Both children do not suffer? No. Son suffers from color blindness while the daughter does not suffer from it. They have a son and a daughter. In this context, which of these statements are most probably correct? Okay, so yes, the last statement, son suffers, daughter does not suffer. Option D is the correct answer. Okay, is this fine? Okay. Now, we're finished with the disease, with the defect part. As before we go into more technologies, yeah. Color blindness is a genetically linked disease. ठीक है? If the child has to be color blind, तो उसका father तो definitely color blind होगा. Right? क्योंकि देखो ये सिचुएशन है, ठीक है. इसमें अब कॉम्बिनेशन बनेगा या तो एक्स एक्स, ठीक है, या एक्स वाई, राइट, गर्ल बॉय. राइट. कलर ब्लाइंड अगर हमने इनको बनाना है, ठीक है, दोनों बच्चों में से किसी को भी या दोनों को, राइट, तो फीमेल के लिए दोनों जीन्स डिफेक्टिव होनी चाहिए, मेल के लिए ये एक एक्स क्रोमोसोम डिफेक्टिव होना चाहिए. इसका मतलब ये है कि यहां से ये सिचुएशन देखो आप, राइट, इसमें क्या हो सकता है कि वन एक्स क्रोमोसोम इज कमिंग फ्रॉम द फादर, दैट मीन्स इट हैज टू बी डिफेक्टिव, क्योंकि इसको तो दोनों डिफेक्टिव चाहिए. दैट मीन्स अगर फादर का एक डिफेक्टिव हुआ तो फादर कलर ब्लाइंड हो जाएगा, राइट. मदर कैन बी इन अ हेट्रोजाइगस फॉर्म, ठीक है, सो मदर हो सकता है कलर ब्लाइंड ना भी हो, राइट. सो देन दिस कॉम्बिनेशन इज पॉसिबल, राइट. इन दिस केस देर इज, अच्छा फादर नहीं भी होगा तो भी अगर मदर भी होगी सिर्फ, ओके, हाँ ये भी पॉसिबिलिटी है. देखो इन दिस केस द मदर हैज टू बी कलर ब्लाइंड और, हाँ एक्चुअली यू आर राइट, ये कई बार सॉल्व करके ही पता चलता है. सो इफ इन दिस सिचुएशन you can have a color blind male with both the parents not being color blind, right? Okay. All right. So we're going to talk about the reproductive process a little bit more. So we have studied this already, that there is an egg, there is a sperm, they fuse, you get a zygote. This zygote then develops into an embryo, and the embryo finally develops into a baby. This period takes time, which is called the normal gestation period. Like for human beings, we say it is 9 months or 40 weeks. Okay. Now, sometimes what happens is there can be variations in the zygote stage. कैसे? जैसे ये zygote बना है, and this zygote sometimes can form another copy of itself. ठीक है, तो इसी zygote जैसा एक और zygote बन गया. In this case, what is going to happen is there are going to be both of these zygotes, they can develop into embryos and then into two babies, what we call identical twins, because they are formed by the same zygote, they will have the same genetic material. So यहाँ पे जो 46 chromosomes हैं, वो दोनों में बिल्कुल सेम होंगे. Just that zygote after being formed creates another copy of itself. The starting point is the same egg and sperm, same zygote, but just that it creates another copy. These identical twins will look alike, will have all the features the same. जो अगर उनकी जीन में कुछ डिफेक्ट है वो भी सेम होगा. Everything exactly will be same for them. However, there is another variation in this where you can have non-identical twins. In a normal process, there is only going to be one egg at one point of time. In the female body, at one time, in the normal course, there is only one egg. But sometimes, under some special conditions, you can have more than one egg or that one egg might split. In any of the cases, if you have two eggs available, sperms are available in millions. Both of these eggs, if they get fertilized, we can get two zygotes. These two zygotes are going to be the non-identical twins. Twins which don't look alike, twins which will be as closely related as the siblings born in different births. ठीक है, जो दो siblings हैं born to the same parents, the kind of commonality they have, these identical twins will also be like that in terms of appearance, in terms of their features, everything. The only thing is they are born in the same birth, but because different egg and sperm are involved, their genetic material will be different. ठीक है, एक टाइम पे एक ही एग प्रिपेयर नहीं, हाँ एक ही एग होता है नॉर्मल सिचुएशन में, राइट, बट नेचर में देयर इज नो सिस्टम विच इज परफेक्ट, सो यू कैन हैव टू एग्स ऑल्सो. So there are two ovaries. At one point of time, only one ovary releases the egg. But under special situations, sometimes both the ovaries can give out an egg. That's when a non-identical twin will happen. So twins who don't look like each other, or when there is a boy and a girl, right? That is a case of a non-identical twin. Okay? Okay. Now, another point into this reproductive process is that sometimes this process does not take place normally. There is some problem. ये जो fertilization है, this is supposed to take place in the female body. That's where the egg is, that's where the sperm will come, zygote वहाँ बनेगा, embryo वहाँ develop होगा, and the female body is going to give birth to a baby. But under some special conditions, this might not be happening. Like maybe the ovaries are not working properly, maybe the uterus is not strong enough to support it, conception is not taking place, anything. In any of those situations, we have the possibility of doing this process in the lab. That process we call in vitro fertilization. You would have heard about this, IVF, or it is also called test tube baby. So in this situation, what happens is, so first, after all the basic tests and diagnosis, the ovaries are stimulated by giving some hormones so that an egg is produced, a proper egg is produced. Then a medical professional can remove this egg. Egg retrieval. Sperm retrieval is easier, that can be done. And both these eggs and sperm, they are brought in the lab. They are given the most ideal conditions of temperature, of nutrients, of hormones, whatever they need, and fertilization would take place. Fertilization taking place means the zygote is formed. And this zygote, when it is successfully formed, it will be stimulated to form an embryo. And once there is a successful embryo, it is transferred back into the female body. Okay. सिर्फ fertilization lab में हो रही है या test tube में हो रही है. The baby has to still go and develop in the body of a female, in the uterus. अभी तक ऐसी कोई टेक्नोलॉजी नहीं है कि टेस्ट ट्यूब बेबी टेस्ट ट्यूब से बॉर्न हो जाए. Okay, that is not the case, right? Test tube baby may only the fertilization would take place in the test tube. Rest of the things are completely normal. Rest of the things are completely going to be the same. There is no difference which is going to be there in this baby or the baby formed by natural conception. सिर्फ उसका fertilization अलग तरीके से हुआ है. At this stage, when the embryo is fully developed, it is supposed to be transplanted into the uterus. And then after normal gestation period, the embryo will develop into a baby and a baby is going to be born. Now here there is a choice. This transplantation could either be done in the biological mother. The biological mother would be the one who has given the egg or the 23 chromosomes. Or there is an option, suppose there's something wrong with the mother's body, she's not healthy enough to carry a baby, or any other case, maybe of a choice also. We can do this with the help of a surrogate mother. A surrogate mother here is any unrelated female. She is going to be holding this developed embryo. She will give birth to the baby. She will provide nutrition to that baby for 9 months. And apart from that, she will not have any link with the baby genetically. Genetically, the baby's composition, the baby's features are going to be those of the people who have given him the egg and the sperm. Because we get our properties from our genetic material from our 46 chromosomes. So, surrogate ki body mein baby will live for those 9 months. Baby will be born through her, but none of her features will be reflected in the baby. Her job is only to give nutrition. ठीक है? The concept of surrogacy is now very widely discussed. You might have come across that in different movies also. Like you had this movie Mimi. Yes? So in that, what happens is that there is an Indian girl who gives birth to a Caucasian white child. Right? That child is white and he doesn't look like that girl or anybody else in her family, but she's ultimately stuck with the child. She's supposed to raise the baby, right? So there is this challenge. Situations like this are very commonly happening in our country. We have a very big surrogacy industry in India. As such, medical technologies are very cheap when it comes to India. The cost of our treatments are very much less, very less as compared to other parts of the world. So a lot of people come to India for treatments. So we have that idea of medical tourism going. The other thing is that we also have, as far as surrogacy is concerned, we also have a lot of people who are available to become surrogates at a very low cost. So that option is also available because of the conditions of poverty in the country. We have many places where, like Gujarat mein Anand is very famous for surrogacy, and there they have, the entire village whose main occupation is surrogacy. Everybody, every household, what they are doing is they are undergoing multiple births and they are becoming surrogates and they take money for that. So the government has recognized that there are a lot of challenges in this process and it's not ideal, it's not very good for their health. So what is being done is that they have banned commercial surrogacy. So there is a Surrogacy Act in India, which you need to know about it. There was recently an amendment also to it, so it stays in news. There are challenges to surrogacy in terms of the economic aspect of it, in terms of the health aspect, the societal aspect of it. So currently the law states that only close relatives with altruistic intentions, they can become surrogate. Because of course surrogacy can be an answer and a very good solution for people to have biological children when it is not happening naturally for them. Okay. But commercially it cannot be done. You cannot take money to do that. That's the point here. Okay, so just go through this act and get an idea as to what is happening. Okay. Okay. Now after this, okay, we will be coming to other technological applications. Let's take a break here. This is going to take a lot of time. So we'll take a break and we'll have to take one more class. So maybe we'll have a shorter 2 classes, right? Because otherwise 1 घंटे का बच जाएगा. So we'll be stuck. So let's take a break here. Now we had studied the structure of DNA, and in that we saw that DNA is made up of a phosphate group, a sugar, and a nitrogenous base. Now, universally, DNA is going to be this only. चाहे DNA बैक्टीरिया में है, चाहे अमीबा में है, चाहे हनी बी में है, चाहे किसी प्लांट में है, wherever there is DNA, chemically its nature is going to be the same. And because of this property, we have the opportunity, we have the option of combining DNA from different sources. We can take DNA from one organism and we can combine it with the DNA of another organism. And when we do that, we call that recombinant DNA. If you just look at this is the most simplistic explanation of what recombinant DNA would be. So you're taking a DNA fragment from one organism. This is the yellow organism. And you are taking another piece of DNA from another organism. This would be blue. The color difference is indicating that these are two different organisms. You can bring these two pieces and you can combine them. When they are combined, we get what we call recombinant DNA. The word vector is used here. The vector here is like when we studied diseases, we use the word vector there, right? Vector is for transmission of diseases, like Anopheles mosquito for transmitting Plasmodium. यहाँ पे vector is also the carrier in that sense that it is carrying this piece of DNA. So DNA carry करने वाला carrier vector. We are going to apply this. We are going to put an example into this so that this whole thing and why we would want to do something like this, that will become clear. Then we'll first let's look at the example. Then we will write the steps of recombinant DNA technology. See here. We have studied about hormones and we said that hormones and their amount in our body is very important. So suppose you need any hormone, there is a hormone deficiency in a person, the person will have to take a hormone from outside. Now this is a method in which we can prepare that hormone. The hormone which they are going to be preparing here is the human growth hormone. Okay, so we are going to see how that can be done. For all products, all things produced, created in our body, the information for that is present in the genes. Okay, this we have studied. So, if you have to produce human growth hormone, the starting point of that will be you go to a human cell. Should I continue? Okay. So there is a human cell. In that, there's going to be a nucleus. Nucleus is the location where all your genetic information is present. And here, from this, you're going to take out the gene which is coding for human growth hormone. अगर human growth hormone बनाना है, then we need the gene for human growth hormone. That is removed, and a gene, see, we can create DNA in the lab, we can make copies of DNA, we can make copies of genes in the lab, but we really don't have the option of just sending one gene and it functioning and producing something. The gene has to be a part of another piece of DNA. So isko carry करने के लिए we'll need another piece of DNA. Now we can get this from any organism, but our choice is to get this from a bacteria. Why bacteria is because bacteria से easier है to get extract the DNA. Bacteria are small, they'll grow easily, it'll be easier for us to manipulate them. So our choice of vector is a bacteria, and bacteria carrying this special genetic material, a small circular piece which is present in bacteria, it is called plasmid. This is a very ideal vector for us because this can be easily removed, easily cut, easily manipulated. So you take this plasmid out, you cut it open, and then you will add the gene for human growth hormone in it. Why this is going to combine or why this is going to join is क्योंकि chemical composition same है. वही phosphate, sugar, nitrogenous base is there in the human DNA. The same thing is there in the bacterial DNA. So they will both join, you will get a recombinant DNA. This is a recombinant DNA because one part of this has come from human beings and one part has come from the bacteria. Now you can put it back into the bacteria and you can make multiple copies of this bacteria. In this situation, the bacteria is containing the gene for human growth hormone. So the bacteria will be producing human growth hormone. Okay. Even if this is bacteria, but because the information which it has received is from a human cell, this bacteria will be producing human growth hormone, and we can create a large number of such bacteria or copies of this bacteria in the lab, and we can extract growth hormone, and that can be sold to any person who is in need of human growth hormone. In this process, what we have done basically is that whatever we desire, whatever product we want, we can just take that gene, add it to the bacteria, add it to a vector, and then we can produce that product. ये तो एक example है human growth hormone का, but otherwise we can do it for any product, anything, any hormone, any enzyme, any chemical, anything which we want to produce. Let's see the specific steps in this process. You can write this also. Recombinant DNA technology. First step is going to be isolation of desired gene. Now see, this desired gene is not a technical term. This is just the term which I am using. कि आपको जो product बनाना है, whatever your desire is for this whole operation, you can pick that gene. जो भी बनाना है उसका gene पहले isolate करना है. That's going to be the first step. Now for isolation of this gene, you will need DNA cutting enzymes to make sure that that individual gene is going to come out from that whole DNA in the nucleus. You will need a DNA cutting enzyme. These enzymes are called nucleases. This is a standard term. When you read your current affairs, you'll read about many other types of cutting enzymes or many other types of nucleases, like side-directed nucleases, endonuclease, exonuclease, right? Without going into any confusion of that, just remember जहाँ word nuclease आया है उसका मतलब ये DNA cutting enzyme. Nuclease means DNA cutting enzyme. Now you've cut and isolated this, so you need now a vector. Vector is DNA which will carry your gene, the desired gene. Vector is that DNA which carries the desired gene, and the example which we have used here, plasmid, which is a small circular independent DNA found in bacteria. Actually, viruses also can be used. They also have a similar composition. They are also very ideal vectors. So here only I will write virus also, so that you can remember. Bacteria or virus, they can be used. Now after you have your vector and your desired gene ready, recombinant DNA can be formed. By joining of desired gene with vector DNA. Now for these two pieces of DNA to join, we also require a joining enzyme. Just like we had a cutting enzyme, we have DNA joining enzymes, and that is called ligase. So cutting के लिए nuclease, joining के लिए ligase. And once this works, recombinant DNA is inserted back to get the desired result. Is this fine? Do you understand what happened here? We're going to do one application of this. One more application. One application we've seen that we can get some products. We are going to see how this can be used for making vaccines. And here we will take help of a viral vector. And this is a category of vaccines which we are talking about. Viral vector vaccines. Your Covishield, COVID-19 vaccine, Covishield. Sure you know about this. A lot of you might have been vaccinated with this also. Then Sputnik 5, they are viral vector vaccines. So when we spoke about DNA, RNA vaccine, there also we spoke about the vaccine making process. I'll just review that for you. For the vaccine to work, you need the antigen of that pathogen to be present to stimulate the B cells so that they can produce memory against that particular pathogen and they can provide protection in future. Okay. So here, however, we are going to be using a recombination. See, whenever you are talking or thinking about recombination, think about this. कि आपको कोई एक चीज चाहिए, there's one particular thing, one particular feature you want, that is going to be your desired gene, that is going to be your DNA fragment. To carry that, you'll need another piece, and then they'll join and form recombination. This is exactly what you're going to apply to make this vaccine also. So what you will have is, again, let me write the same term so that it becomes a little easier for you to correlate. So there's going to be a desired gene which is going to be combining with a vector. ठीक है. इस केस में अगर वैक्सीनेशन करना है या वैक्सीन बनाना है, so our desired product is the antigen of COVID-19. Okay, once the antigen is produced, then that whole thing will start working, the B cell stimulation and everything. So our desired gene has to be the gene which will produce this antigen, which has the information for producing the COVID-19 antigen. So we are going to get this gene from COVID-19. So here the desired gene will be the gene coding for or having the information for COVID-19 antigen. ठीक है. Now the choice of vector. In this case, we have to find a vector which does not have its own antigens. That is very important. We want to create protection only against COVID-19 antigen. If you choose a vector जिसमें अपने भी antigens हैं, which is also stimulating the B cells, the immune response is going to become divided. We want that this immune response of B cells, where antibody production would be taking place and memory would be created, that should be limited to COVID-19. So vector हमें ऐसा चाहिए जिसको B cells पहचाने ही ना. Okay, we need a vector to which B cells do not react, do not respond. And that vector we find in a virus called adenovirus, which we pick up from the chimpanzees. Now there are many varieties of adenoviruses, but some of them cause human diseases and some of them the human body does not respond to. So common cold adenovirus se hota hai, but we are not using that adenovirus. We have brought an adenovirus from chimpanzee. This is the chimpanzee's body is responsive to this virus, but in human body there is no reaction to this. Human body इसको antigen नहीं मान रही है, harmful नहीं मान रही है. There is going to be no human immune system response to this virus. If this virus goes into the human body, B cells do not recognize it. B cells don't have any action against it. The immune system is not stimulated. This is essential so that the only response which we get would be against the COVID-19 antigen. Okay. So this vector we take, and then when these two join, you get a recombinant adenovirus with gene coding for COVID-19 antigen. This whole thing is then, the virus is weakened because that's how you deliver a vaccine. So this whole thing is converted to a live attenuated form and then delivered as a vaccine. Now, when this weakened virus is going to go into a person's body, the B cells will start checking that, okay, let me check which antigens are there. What work do I need to do? And adenovirus के अपने कोई antigen नहीं मिलेंगे उसको. The only antigen B cells will be able to detect will be the ones which are coding for COVID-19. COVID-19 antigen is going to be produced. B cells will recognize them. B cells will get stimulated. They'll produce antibodies against COVID-19 and they will have the memory for that. That is ultimately our objective in this process. We create a viral vector vaccine by taking a vector to which our body does not respond and recombining it with the gene coding for the antigen of the pathogen against which we are seeking protection. Doing this will give us the viral vector vaccine because here we are using the virus. This was the last type of vaccines in that long series of vaccines which we've been talking about. Is this clear? Okay. So there is a question also which I want you to solve on this. Today we are also going to give you a assignment question based on types of vaccines. So to write that question, you will have to go through the vaccine making process, and this is actually a UPSC question which I've slightly derived to make it a little easier for you to write. So do make the effort of doing that. आपको पहले vaccine पढ़ना पड़ेगा and then you will have to write about all the different types of COVID-19 vaccines. So we had one in the inactivated killed variation, Covaxin. Then you study DNA and RNA vaccines, and the fourth type is going to be this one, the viral vector vaccines. You'll have to write about all these four types. Sudarshan, please find the question. There's a question, how vaccines work. ठीक है, आपको मिल जाएगा अपने डाटा में, and we have to give that as an assignment question. Sorry. Okay, so Mohit says the answer should be D. C, C, D. Recombinant DNA technology or genetic engineering allows genes to be transferred across different species of plants, from animals to plants, from microorganisms to higher organisms. Now, there is no difference between the DNA of any of these organisms. ठीक है, चाहे microorganism का, चाहे higher organism का, plants, animals, different species. For all of these, the genetic material is the same. DNA का composition design सब same. So, genes can be transferred across all of this. Extra दिमाग बिल्कुल नहीं लगाना है कि ये क्या बात हुई, ऐसे क्यों लिखा है animals to plants, right? Just because it doesn't appeal और आपको सुनने में अच्छा नहीं लग रहा है, that cannot really be a logic for finding the answer. The logic has to be in the fact same DNA plants में है, same DNA animals में है, it should be transferred. हाँ, if you think further, has this been done? Yes, all of this has been successfully done. Is there any use of doing this? Yes, that is something which can be questionable. ठीक है कि इसका कुछ useful product बनेगा या नहीं बनेगा. Okay, but that's really not what the question here is dealing with. Can it be done? Yes. Can the transfer be done? Yes. Has it been done? Yes. All forms of DNA can be combined because DNA is the same everywhere. So answer is going to be D, 1, 2 and 3. Okay. Now the next thing which we are coming to here is the fact that like suppose somebody has a deficiency of growth hormone. So हमने recombinant DNA technology use की, growth hormone बनाया, उसको देना शुरू कर दिया. ठीक है, the example which we just studied. Now think of a situation where we know that this gene defect is because, this lack of growth hormone is because of a gene defect. Because of a gene not working properly, the gene producing for growth hormone. If instead of regularly giving the person growth hormone, what if we can give him that functional gene? अगर उसकी जीन काम नहीं कर रही है, अगर हम उसको जीन न्यू दे दें, then he'll never have to take human growth hormone from outside because then his body will be equipped to produce the human growth hormone. Now we can do that, we can make such changes, and that process is called gene editing. Gene editing or DNA editing is basically a process in which we can add, replace or delete genes. We have technology now where there is this possibility कि हमें जो gene remove करना है, add करना है, replace करना है, we can easily do that from the genome of a person in the person's own genetic material. And let's see how that will work. First we'll understand it simply, then we'll talk about the specific technology. Just look at this image. The genetic material is present in the cell in the nucleus. Okay, the nucleus is the location of the genetic material. Now this is the part of your DNA, of the genome, which is supposed to be, say, removed. Okay, say there is something wrong with this. Suppose this is causing sickle cell anemia in the person, right? So we want to remove this gene. The first step here would be to prepare a guidance molecule to identify this location. DNA as such is a very large molecule. So आपको एक जीन तक पहुंचने के लिए you need something to just take you to that location. So for that you have guidance molecules. These we have created technology now where we can program this. We can create different types of guidance molecules based on which gene we want to reach. Along with this guidance molecule will come a DNA cutting enzyme. So molecule वहां तक ले जाएगा, फिर DNA cutting enzyme. What is the name of the DNA cutting enzyme? Nucleus, right? So this is a nucleus which is going to cut DNA at this particular site. Once it is done, you can allow the cell to repair on its own, or if you want to add some other gene, you can add that gene. If you want to keep it deleted, you can keep it like that. Once it is open, you have all the options with you. ठीक है? The thing here is that for this to work, a guidance molecule chahiye, a DNA cutting enzyme chahiye. Now, there are many ways in which gene editing was tried out, but this method is the most successful method. This is also a Nobel Prize winning technology, and this is called CRISPR. Please write down its full form. This is clustered regularly interspaced short palindromic repeats. In short, CRISPR. Now, this technology or this kind of operation was naturally done by bacteria. Okay. This we have developed from bacteria into a technology which will help us in gene editing. CRISPR is called gene editing tool or molecular scissors. The two parts here are going to be, first is the guide molecule. The name of this guide molecule is single guided RNA or sgRNA. This is also the exceptional RNA in the sense that it is double stranded. The second component here is the DNA cutting enzyme. The DNA cutting enzyme is the nuclease and its name is Cas9 protein or Cas9 enzyme. If you remember, all enzymes are proteins. We studied this when we were studying proteins. So the name of this is Cas9 protein. So what is happening here is this we can create, sgRNA we can create based on which gene we want to attack. Okay. And this is going to take the whole system to the location where the Cas9 is going to cut it. If you want, you can write one line also. Please write. Single guided RNA can be created for identifying specific parts of the genome, and the DNA cutting enzyme Cas9 protein, Cas9 protein cuts the DNA at that specific site. At that specific site. Okay. Now think a little bit about this technology. Okay, we have technology where we can identify any part of the genome, identify any gene, and we can cut it. After that, we have the option of changing any gene, removing any gene. For different kinds of gene defects which we have studied. Can you tell me an example of a gene defect? Turner's is not a gene defect. जिसमें individual gene defective होती है, that kind of a disease I'm asking. Not Down syndrome. Down syndrome, Turner's, the defect is of the whole chromosome, not of one gene. अभी वो ही बच गई हैं, अभी वो क्रोमोसोम वाली तो बोल ही दी आपने सारी. तो it was obvious that's where you heading. Yes, very good. Sickle cell anemia, hemophilia, color blindness. जहाँ आपने लिखा था कि these are gene defects, that was the question. Okay. So when individual genes are defective, such a treatment can be a very good solution for diseases which otherwise have no solution. Now CRISPR has recently been approved in UK to be used for sickle cell anemia. They're starting this as at an experimental level. Okay. To do this, one thing you have to consider is that if you want to have the best impact of a gene editing, it has to be done at the early stages of life. Because you will need that change in all the cells. अगर आप later on करोगे, कुछ targeted locations पे करोगे, you might need to do it again and again. Once the new cells are formed, they'll again need those changes. So, if you can do these changes in the embryo where the number of cells are less, or even before the embryo, जैसे egg और sperm है, like the cases we were considering, suppose both parents are colorblind, right? तो अगर आप egg and sperm में ऐसी editing पहले ही कर दो, you will make sure that the child will not have color blindness. You can remove that defective gene. Okay. So this gives us a lot of potential and a big solution for diseases about which you cannot do much once the person is born, or later on any other form of alternative treatments is not there. However, the consideration is this, that there is no limitation to doing this only for defects. ये एक editing technology. आपने अगर सीख लिया कि ये जीन आइडेंटिफाई करके कैसे रिमूव करना है, you can apply this for anything which you want, for any character, right? Suppose I am the autocratic ruler of a country and I decide everyone in my country must have curly hair. Okay, so the moment an embryo is conceived, I am going to do gene editing in that embryo and all children will be born with curly hair. Because I will introduce that gene for curly hair. Okay. Now this is a change which is not really required, right? This is not clinically indicated, this is not a disease, but I have the technology, I have the money, or meri chal rahi hai, to I can do whatever I want. The argument against this kind of a technology is the concept of designer babies. See, CRISPR was given the Nobel Prize in 2020. And the big thing about CRISPR was that this was in chemistry. Okay, this was given the Nobel Prize for chemistry. It was the first time that 2 women received a Nobel Prize for science together. Okay. Now, they have been building this technology for many years. Okay, UPSC में भी already 3 question आ चुके हैं उससे बहुत पहले. Right? This has been in place. But abhi bhi its use is mostly experimental. This is the first disease, sickle cell anemia, for which it has actually given uh, clearance ki okay, try karo, use karo. Abhi bhi try karenge, then they'll come to know ki okay, what is happening, what are the challenges they are facing. But the idea or the reason why there was a moratorium, or why they felt that it was important that they need to take some time to understand the impacts, is because of this issue, because of the idea that there can be a very big potential of misuse of this kind of a technology for anyone who will have resources. People might start going in and saying कि ये 5 genes मेरे embryo में डाल देना, these are the features I want in my baby, whatever that is, right? And at the level of nations also, there can be some very unfair advantages which a country might have, right? Like, China, right? Actually, you don't know China, nobody knows China, nobody knows what goes on in China. But again, time and again, some report, something keeps coming up कि China में ये हो रहा है, China में वो हो रहा है. So one of those reports which come is that China is doing some gene editing, and China is creating a special breed or special variety of soldiers. They are preparing their soldiers with special characteristics where they'll have whatever traits you need in the army, like better muscle mass, better strength, better agility, whatever is needed. So this, in terms of the technology which we have, the potential is there, वो कर सकते हैं. But आप सोचो इसका कितना unfair impact हो सकता है, and how does, the bigger question is, how will it impact gene evolution in future? How will progression of human race go ahead if we keep doing these changes? Because of these concerns, there has been a very cautious decision to slow down the use of this technology, which can actually solve so many problems, and which is a solution which we've been waiting for different gene defects. Okay. If this is something which interests you, you can listen to this TED talk also. Do you know about TED talks? Yes. So the person who has received who has created this CRISPR, so her name is Jennifer Doudna, and I mean one of the persons. And Jennifer Doudna का एक बहुत पुराना TED talk है on CRISPR. So if you search CRISPR on TED talk, you'll find that it's a 15-minute talk. She explains how they identified this, ये कहाँ से ढूंढा, कैसे idea है, and she explains a great deal about what are the concerns of why they don't want to go ahead and roll this out in a big way. ठीक है? Okay. So here, as we progress more towards mains kind of topics, so technology part, yeah, how it works, that becomes more of a prelims oriented thing. In these sections, you'll need to think about all these ideas also. Iska kya advantage hai, iska kya disadvantage hai, what are the challenges, why it's not being used, why it should be used. Because these are the questions which you will get in the mains part. And you'll not really get a question to explain a technology. Maybe they'll ask you what a technology is, but जो technical steps है, इस type के questions नहीं आएंगे. Application based questions they're going to be asking. So you'll need to think about all these things. Okay. The next topic which we are coming to is this. Do you know who she is? First clone. Yes. So this is Dolly the sheep, and this is the first successfully cloned mammal. Okay. Now we are going to understand what is cloning. So here we have, let's talk about the normal reproduction process first. So we've talked about this so many times now. When a zygote is formed, it has 46 chromosomes, but these 46 chromosomes have been equally contributed by the two parents. 23 mother ने दिए, 23 father ने दिए. That is why the zygote and hence the child which is going to be born is not going to be exactly like either of the parents. It's going to be somewhat of a mix, right? How your parents keep reminding you when you do something wrong, ki mummy pe chala gaya ya papa pe chala gaya, right? The idea of that is that you have traits of both the parents because both of them have contributed to your genetic material. Now, cloning would be something which does not follow this rule. Animal cloning means that you create an exact copy of a person. So that means in this case, there's going to be a single parent. If we create an exact genetic copy of an organism with all the same features, same chromosomes, like agar human being ki baat karo, same 46 chromosomes, that is going to be a clone. ठीक है, जैसे कि हमने यहाँ पे ट्विन्स की बात की थी, ये वाले आइडेंटिकल ट्विन्स. So identical twins are the natural clones. Why they are the natural clones? They have the same 46 chromosomes, same features, everything about them is the same. Now we have developed technology to do something like this in the lab. For creating a clone, we are going to have one parent who will be the only contributor to genetic material. Okay. Now we have two types of cells in every organism. One is the gametes, जिसमें है n number of chromosomes, and you have the somatic cells. Remember somatic cells? With 2n number of chromosomes. Okay. So other than gametes, all the cells in your body are somatic cells. And this is the complete set. So for this kind of reproductive process or in cloning, we cannot really use the gamete. क्योंकि gamete के पास आधा ही है. How the functioning of the gamete works is both the parents are contributing this n. Then only we get 2n. पर यहाँ पे एक ही parent है तो 2n इसी से लेना पड़ेगा. 2n is present only in the somatic cell. So the starting point of this process will be a somatic cell. We'll start this with a somatic cell. So you can keep writing this in the form of a flow chart so that you remember the steps, what is happening here. So you have a somatic cell with 2n number of chromosomes. This you have taken from the organism which you are trying to clone. From the person who is going to be the parent, whose copy you want to create. Okay. But what happens is, in nature, for the process of reproduction, for your zygote to change into an embryo and then develop all those supporting features to get nutrition, an egg is essential. Egg is the only cell which has the property to make an embryo, to make all the other structures like placenta and umbilical cord या जो भी और चीजें चाहिए. So for reproduction, for creation of a new organism, you will need an egg. Okay. So एक parent है, egg आप किसी और से ले सकते हो. You can call that as the donor. Egg has n number of chromosomes. ठीक है. Now, if you are trying to make a copy of this person, you only want these chromosomes in the nucleus of the somatic cell. ये जो chromosomes egg से आ रहे हैं वो नहीं चाहिए. We want the properties of the eggs but not these chromosomes. So we will follow a process called enucleation. Enucleation is removal of nucleus. In both these cases, we are going to remove the nucleus. With that, what will happen is, we'll keep the egg without the nucleus. So we are calling it the empty egg because no nucleus is there. And from the somatic cell, you're going to keep the nucleus. So we have somatic cell nucleus. So जहाँ से हमें characteristics चाहिए, जहाँ से हमें chromosome चाहिए, we've kept that nucleus and the other nucleus we have removed. Now what we can do is, we can do a transfer. This is called somatic cell nuclear transfer. And what happens as a result is, this is the nucleus of the somatic cell and this is going to come into the egg. So this is now egg with somatic cell nucleus. This is now ready. It's just like the zygote. Zygote may be 2n number of chromosomes there with all the properties of the egg. This is similar to that. So this is ready to divide and create an embryo. In the lab, as we are doing this process in the lab, what we can do is, this can be stimulated to form an embryo. This embryo can then be transplanted into the uterus. Once you have a successful embryo, it can be transplanted into the uterus, and with that, this embryo will develop into a baby. You will get a clone who is going to be exactly like the person or the organism who has given the somatic cell. Because this clone has the same 46 chromosomes, will have all the same properties, will look alike, everything will be the same क्योंकि genetic material exactly same. If I apply this and review this again for you, suppose you decide that you are going to clone Virat Kohli. ठीक है? So Virat Kohli को अगर clone करना है, the starting point of that will be that you get a somatic cell. This somatic cell you will get, that means उसका sperm को छोड़ के any other cell from his body you can use. Now you want all his properties, all his 46 chromosomes, so you are going to remove and keep his nucleus, the somatic cell nucleus. You need another person, you need a female who will donate an egg. So maybe you can ask Anushka, we can use one of her eggs, right? But we don't want any of her properties, right? Because then the baby will not be Virat Kohli, but will be a mixture of both of them. So इसका nucleus नहीं चाहिए, इसके 23 chromosomes नहीं चाहिए. We'll remove this, we'll just take the empty egg, we'll do a somatic cell nuclear transfer. Once the embryo will be formed, it can be transplanted back into the uterus. This uterus can be of the same female or of another female. That will not make any difference because that is just for providing nutrition. The genetic material of this baby is coming from the somatic cell. That means only from Virat Kohli. Now this baby, once it completes its gestation period, it is going to be born. It will look like Virat Kohli. It will have all his properties, everything which is genetically controlled. Okay, even if he has some diseases which is having some genetic basis, the baby is going to have that also. Okay, of course, there's going to be a difference in the age, right? The baby will not be born with a beard. Okay, those variations will be there. And depending on how this baby is raised or what kind of environmental variations are there, that is also going to be making a difference. You can't raise this baby in Russia and then say कि चल अब पंजाबी में गाना सुना और cricket खेल के दिखा. He might not know that, right? Because that is something which is environmentally ingrained. But other than that, how he looks, how his body is, everything is going to be exactly the same. Okay. Now, as of today, in the world, anywhere, other than I think science fiction movies, human cloning is not allowed. Okay, this is something which is not legal anywhere in the world, except for again rumors like when Osama bin Laden died. Uh, so there were these reports that the Navy SEALs got the wrong one. So there is a real Osama bin Laden, and he has 4 clones, and they killed the wrong one. The real one is still alive, probably hiding somewhere in Pakistan. So other than all these stories, it is not allowed anywhere. Let's see what happened with Dolly. Same process exactly. So Dolly, when she was cloned, this is the sheep which was used for cloning Dolly. And in this case, the somatic cell was taken from the sheep's udder. Udder is basically the milk glands which are there. So from that, one somatic cell was removed, and then the nucleus of that is going to be kept, because that's where the genetic material or all her chromosomes are there. Then another sheep was brought in for the egg. This could be, the nucleus would be removed and just the empty egg will come to hold the nucleus. You will get the complete cell with the nucleus with the complete set of chromosomes. This is stimulated, it forms an embryo. Embryo is put back into a sheep and a lamb is born. The cloned lamb has the same features, thus everything same as the sheep who has donated the somatic cell. Animal cloning, however, is permitted. Animal cloning is done also. Even in India, we have cloned almost all our domesticated animals. So cow, buffalo, camel, goat, sheep, हमने सब किया है. ठीक है. The question which I want to ask you is, why would we want to do this? What can be the benefit of cloning? Okay, so for some desirable features, okay, like suppose there is a cow which is some exceptional variety giving 100 liters of milk. Okay. Now we cannot afford to lose this magical cow, right? It's giving 100 liters of milk in a day. So we can make a clone of this cow, because if this cow will reproduce normally, so वो जो दूसरी 5 किलो वाली cow है उसकी properties भी आ सकती हैं, right? The second parent. So in order to preserve such exceptional desirable characteristics, we'll make a clone. Clone में उसी का copy बन जाएगा. Everything, all the properties are going to be perfect. ठीक है? Anything else? Any other use you can think of? सोचो थोड़ा सा. Ma'am, how mitochondrial swapping deals with genetic diseases? We'll study this in the next class. See, we can use this one very good application could be for organisms who are endangered, whose numbers are decreasing and they're not able to produce naturally, or sometimes there is a situation that, the sex ratio is not normal, that there are no males left or no females left. So in that case, these kind of options can help in increasing the number of those organisms. Okay, and clones can be a very good way to for any kind of organ transplants or blood matching or anything which is needed for a species. So for that also we see a lot of applications. In theory, this is a perfect system. But in reality, what we have observed is that these clones do not have a very good quality of life. Like Dolly developed a liver disease and then she died after some time. In India, our experience has been that we have seen clones being born and dying within months and days also at times. Okay, so we don't understand a lot of this, that why this would be happening. I think the closest explanation would be something that, you know, we can try everything perfectly, but then nature has its own rules and it's powerful than everything else which works, right? So we've not had a lot of success in that sense. We have been able to produce animals, but not with very healthy animals which have a long life. Even this is true for every time we use some assisted technology like IVF. Okay, we spoke about IVF. IVF is done in the most perfect conditions, but in that also the success rate, realistically speaking, is close to 15%, because there is a chance of things missing out at different stage. That is why what they do is they give a lot of ovarian stimulation to produce multiple eggs. So they'll give a lot of hormones so that a multiple number of eggs are produced. And in that case, if suppose 2 eggs are produced or 3 eggs are produced, jitne bhi eggs banenge, all of those are going to be retrieved, and they will try and fertilize all those eggs. Okay, sperms as such are available easily, right, in large number, in millions. So that's not a problem. And after that also, if fertilization takes place, if embryos are formed, if suppose 2 embryos are formed or 3 are formed, they will transplant all those embryos, because किसी भी स्टेज पे वो फेल हो सकता है. एम्ब्रियो की स्टेज पे फेल हो सकता है, एम्ब्रियो इम्प्लांट करते वक्त फेल हो सकता है, उसके बाद अर्ली फर्स्ट सेमेस्टर ऑफ प्रेगनेंसी में फेल हो सकता है. So they just want to ensure that, because it is a difficult process, it is an expensive process, that the chances of success should be higher. So people who undergo IVF treatment in this case have a higher probability of having multiple births. Why? Because all the embryos which have found success, they are going to be transplanted. So 2 children are very common in people who have IVF, or even 3 at times. Like, you know, Farah Khan? No? So Farah Khan, 3 children through IVF because of production of more eggs. In this case also, what we see is that, like Dolly ko banne se pehle, there were 200 plus embryos which were destroyed. Okay, so success in this situation also became, is very challenging, is very complex. And for human cloning, abhi though it is not allowed because of a lot of issues which we might have when it comes to identity or other things, or things like, you know, Osama bin Laden escaping or something. But the data which we have, based on that, it hasn't been done, but the data tells us that this would be a very, very difficult birth. It will be a very difficult pregnancy whenever that is done. And there are estimates which say that the size of the head of the baby, is going to be abnormally large. And it can be so large that the womb might actually tear. Again, these are estimates and not something which has been experimentally proven. And we don't know when it could actually be done, because as of now, experimentation and human cloning and all levels is not allowed. If it is allowed someday, it can be a good answer for organ transplantation. It can be a good answer for childlessness. It could be a good alternative reproductive technology. And if you look at this, for females, the interesting thing is that just one female can reproduce on its own without anybody's help, without any other organism or its genetic material being involved. जैसे कि अगर इसमें आप विराट कोहली की जगह अनुष्का का क्लोनिंग करो, ठीक है, सो अनुष्का से सोमैटिक सेल ले लो, एक अनुष्का से ही एग ले लो, you do a transfer, you stimulate, form an embryo, and then it can be transplanted into her body and she can give birth to her own clone without the need of any man. Okay, so that, if it happens someday, might be a possibility. Is this clear? सब कुछ नॉर्मल होगा, एक बार बेबी बॉर्न हो गया, बाद में सब कुछ सेम है, ठीक है. इट्स जस्ट दैट वी आर जस्ट इंटरवीनिंग इन द प्रोसेस ऑफ क्रिएशन, बट इस स्टेज के बाद, once the embryo starts forming, once it is put back into the uterus, everything is exactly the same as it happens in nature. तो टाइम तो दोगे ना आप इसको डालने के बाद, 9 महीने लगेंगे ना, नॉर्मल gestation period जो होता है, that you have to give, right? So there is going to be a full grown pregnancy and then birth. ठीक है ना? और बेबी को भी ग्रो होने में उतना ही टाइम लगेगा, राइट? उसमें भी कोई उसके बाद ऑल्टरेशन नहीं है कि वो अभी 5 साल में ही विराट कोहली जितना बड़ा हो जाए, ऐसा नहीं होगा, राइट? वो सिर्फ मूवीज में होता है कि ऑल द क्लोन्स लुक द सेम एज, राइट? सो हाँ, अगर बर्थ के टाइम ही किसी की क्लोनिंग कर दे, देन दोज बेबीज विल बी 1 year अपार्ट. ओके, यू नो talk about another technology or test which is called DNA fingerprinting or the DNA test. I'm sure everybody has heard about the DNA test. Yes, where is it used? Movies. अच्छा, मूवीज में कहाँ यूज करते हैं, कहाँ एप्लीकेशन होगा इसका? फॉरेंसिक्स, ओके. क्रिमिनल इन्वेस्टिगेशन, व्हाट एल्स? क्या? आई डेंट गेट द जोक. मुझे भी बता दो, मैं भी हंस लेती हूँ थोड़ा. आधार कार्ड. बहुत मुश्किल प्रोसेस है, बहुत महंगा पड़ेगा गवर्नमेंट को, इफ द गवर्नमेंट डज दैट. And there are a lot of privacy issues also on this, that use of genetic material. Okay. So for paternity disputes, this is one method where direct correlation can be drawn. Now, DNA of every person in terms of the sequence which we have, the sequence of A, G, C, जो भी है, right? That is unique to all of us, but we are getting this DNA from our parents, so there are some sections which are exactly going to be the same. These sections are in smaller forms available, which are called variable number tandem repeats. For finding the father of an offspring crimes. Yes. This is called VNTRs. See, we spoke about genome sequencing, right? Where we check the whole sequence of DNA of a particular person. But that is a very difficult process, very time consuming because of the large size of DNA. We cannot do that every time we want to do a DNA test. So we have identified some sequences which are unique to a person and which can be easily recognized, and only that small sequence is going to be, you are going to be sequencing only that small part. So ये जब DNA की sequencing होती है, it's not going to be sequenced as one chain, but you first divide it, you first break it down into smaller parts, and then individual parts are sequenced, and then they kind of create that whole data together, put it together. So instead of doing that for the whole DNA, which is going to be a very difficult process, that's why see, India का जो ambitious project है, that is doing it for 1000 people, right? That's the big project. That's also going to take years. So उसकी जगह ये एक तरीका है where you have sequences which are smaller, they are called VNTRs, they are easier to identify. So a person's DNA can be extracted. सब cells में DNA है, hair से ले लो, blood से ले लो, skin से ले लो, internal organ से ले लो. Anywhere, wherever you have the cell, you have DNA in the nucleus of that cell, and that DNA is the same. Hair cell में, blood cell में, हर जगह वो same है. From that cell, DNA can be extracted, that DNA can be broken down, and in that, these small segments, they can be recognized. these segments are unique for a person and they also match with the parents. So paternity establish kar sakte ho. Relatives will also have some commonality here, like siblings. So there can be a matching between siblings, there can be a matching between parents, even grandparents, because of the same line of inheritance. But this is easier because the sequences here are small. We call this as satellite DNA also. And basically this term repetition you have to understand. Repetition का क्या मतलब है कि या repeats का क्या मतलब है, that some part, some sequence is going to keep repeating again and again. For example, suppose you have T, C, A, G. T, C, A, G. So there can be a sequence which can keep repeating itself, say 10 times, 20 times, 100 times, like that. Now because of this repetition, it becomes easier for us to recognize this part. ये कितने nitrogenous base होंगे इस repeated sequence में, that can be variable. That's what it means, variable number tandem repeats. We have two main types of satellites or two main types of satellite DNAs. One is called mini satellite and the other is called microsatellite. So mini or micro में से which do you think is going to be smaller? Micro. Yes, micro is smaller, mini is bigger. Variation क्या है? Micro is when the number of bases are 1 to 7 nitrogenous bases, and mini is 7 or more bases. So like in this case, you have 1, 2, 3, 4, 5. 5 bases being repeated again and again. So will this be a mini satellite or a micro satellite? Micro. ठीक है, less than 7. So ये जो हैं, these can be identified. They'll give us information of a particular organism. They can help us identify parents because we have these VNTRs which we have common with our parents also. Or say any person has to be identified from a crime scene, or any unclaimed dead body has to be identified. सब में DNA test आप कर सकते हो. Okay. Like there have been so many famous cases of politicians with who had to go through a DNA test because somebody somewhere rose one day and said ki I am your child, no? Like ND Tiwari and Jayalalitha. Okay. So in that case, the VNTR of one person can be matched with that of their parent, and if it matches, you know they are biologically linked. ठीक है. Crime scenes पे क्या होगा? Suppose there's a person who is suspected for a murder, and at that site you have some blood stains or you have hair available जो जहाँ पे crime scene है. So that hair strand can be taken. It has cells. From that cell, DNA can be extracted, VNTR can be identified, and all the suspects, suppose there are 3 suspects, from that they can do a matching from their DNA sample. So जिसके DNA से match करेगा, जिसके VNTRs match करेंगे, that will be the person who was there at the crime scene. So this is a very definite, sure shot test for identification of a person. ठीक है? Okay. So we will be ending our class here. If you want to ask anything, you can please ask. We have a few topics left which we will be doing next time. Whenever we have a class this week only, I think, or no? Okay, whenever that is, then. Try and revise your vaccine process and try and write that answer which you are getting today. So in your class 10, there is this chapter 8 on heredity and evolution. Evolution part you can choose if you want to read or not. Heredity wala aap पढ़ सकते हो. Most of this that should make sense to you. So next class is going to be the last one. So if you have any doubts from any section, you can bring it in the next class. Okay. All right. Thank you. सेपरेट नहीं होते, जॉइंट होते हैं मतलब. हाँ, अब जैसे जाइगोट जब ब्रेक हुआ, ठीक है, सो कई बार ऐसा हो सकता है कि वो पूरा डिस्कनेक्ट ना हो. देयर कैन बी सम पार्ट विच इज गोइंग टू बी स्टिल अटैच्ड ऑफ दोज टू सेल्स. सो वो जहां से अटैच होगा उसने जो पार्ट बनाना था वो पार्ट ज्वाइन हो जाएगा. अगर हार्ट बनाना था तो हार्ट कॉमन हो जाएगा, हेड बनाना था हेड कॉमन हो जाएगा, लेग्स बनानी थी लेग्स ज्वाइंट हो जाएंगी. So it just means that those two embryos, when they were developing, they did not really disconnect completely. और जहां से join हुए वो part common हो जाएगा. We call them conjoined twins. ठीक है? If they have all the parts which are there, they can be surgically separated. But sometimes what happens is they are two twins, but they have one heart. So उस case में they cannot be separated without losing one of them, right? हाँ, आइडेंटिकल नॉन आइडेंटिकल का यही लॉजिक है ना. जैसे हमारे इसमें कहाँ गए मेरे ट्विंस. देखो, जब ये जो हमारे सेक्स डिटर्मिनिंग क्रोमोसोम्स हैं, दे आर अ पार्ट ऑफ दीज 46 क्रोमोसोम्स. ठीक है, सो जब इसी सेम 46 का एक कॉपी बनेगा तो सेक्स क्रोमोसोम्स भी सेम रहेंगे. दे आर अ पार्ट ऑफ दिस. तो अगर XX था तो XX ही आएगा, XY था तो XY ही आएगा, राइट? इन आइडेंटिकल थिंग ट्विंस यू कैन नॉट हैव दैट वेरिएशन. बट नॉन आइडेंटिकल में एग और स्पर्म डिफरेंट है. सो इसमें एक वेरिएशन हो सकती है. एग के पास तो वी हैव स्टडीड it's going to be X chromosome, but the two sperms which are going to come here, one sperm can be carrying an X chromosome, the second one can be carrying a Y chromosome. So one child will be a female, the other can be a male. जहां पे दो एक लड़का एक लड़की है, it is a definite case of non-identical twins. But if there are two boys or if there are two girls, in that case, it can be identical also, it can be non-identical also. You'll have to look at their faces to see if they are the same or not. Okay?